Great. Thanks, everybody. And we'll get, we're the last session between you and lunch, but I think this will be an interesting one for everybody. So I'm going to take just a moment to provide a little bit of context, and then I'll sit down with my fellow panelists who will introduce themselves, and then we'll just jump into the discussion. So we've heard a lot of discussion over the last day and a half here about in many cases, what if we could stop disease before it ever begins? So I think for most of history, we know that doctors have had to wait for symptoms. A tremor, stiffness, only then Parkinson's disease. A child that's rushed to the ER with dangerously high blood glucose, only then type 1 diabetes. By that point, damage is already done. But here's the truth. Those diseases don't start suddenly. Parkinson's can brew silently for years with brain cells dying before a handshake. Type 1 diabetes begins with the immune system quietly attacking insulin-producing cells long before the blood sugar spike. By the time we see it, the damage is already done. And we've lost precious ground. Historically, doctors have been forced to be reactive. They used the tools they had, symptoms, stethoscopes, blood tests, and they arrived after the fire was already burning. Medicine was firefighting and often in the dark. But today, thanks to the molecular era, we have night vision. Genetics, biomarkers, advanced imaging, they let us spot disease in its pre-symptomatic phase. We can see the earliest sparks, and instead of waiting, we can step in. That's interception. So imagine catching Parkinson's a decade before tremors or cognitive loss. Protecting vulnerable neurons before they're lost. Imagine spotting the beta cell problems or immune shifts that lead to type 1 diabetes. Imagine doing this routinely for any chronic degenerative disease. This shift is urgent. These diseases don't wait for us to catch up. Interception is the bridge between prevention and treatment or cure. It moves us from reacting late to acting early when change is still possible. So picture a world where disease is routinely caught and stopped, not in the ER or the hospital ward, but years earlier before it takes hold. That world is within reach, but we lack a clear map of how to get there. In some cases, we've managed to get close. We have some pieces, some of the dots. But these tend to be sporadic examples as opposed to the result of a focused, consistent, and determined effort to make reaching this destination a matter of routine. How can we connect the dots from these examples? How can we come together and create a map, a framework that can be followed by all? This is the challenge that our panelists for this session have accepted, and one that I hope all of you in this room will accept too. So with that, I'm going to have a seat, and we'll start with introducing Carly, and we'll just go down the row.
Say who I am?
Yeah.
Okay. Yeah. So I am a professor of neurology at University of California, San Francisco, and I work in clinical research, clinical trials, epidemiology, focusing primarily on Parkinson's. I also take care of patients.
Hi, I'm Jiri Aubrecht, and I had a long career in the pharmaceutical industry in large companies like Pfizer, Takeda, and then small biotech companies afterwards. And now I am an adjunct professor at Georgetown University.
Kevin Krudys. So I'm the Associate Director of Quantitative Science at the Office of Neuroscience in Cedar FDA.
And I'm Josh Vieth. I'm Senior Director of Research at Breakthrough T1D, formerly JDRF, and I lead our disease-modifying therapies portfolio.
Great, thanks. Josh, I'm going to kind of start with you, and we're hoping to have sort of a conversational interaction. So, you'll see us, ask each other questions, and then at the end, I'm hoping that you from the audience will jump in with your ideas and thoughts as well. But Josh, maybe we'll kick it off with you. Type 1 diabetes, I think, is one of the clearer examples of how we have an opportunity to intervene early. I know Breakthrough T1D has been working on this for literally decades now. Can you talk a little bit about, given that we have insulin, we have marvelous technologies, why even think about intervening pre-symptomatically?
Yeah, and I think it comes back to treating the pathology or biology of disease versus the symptomology. Type 1 diabetes, the symptomology is metabolic. It's glucose control, it's insulin production. But the underlying disease pathology is autoimmune in nature. And we've been treating type 1 diabetes with insulin for over 100 years. And it wasn't until a couple years ago that we had the first disease-modifying therapy for T1D approved, which is a T-cell focused drug, teplizumab. And that is actually in line with this panel for pre-symptomatic patients. So it's in the pre-symptomatic stage of disease. What we can gain from understanding those biology, those symptoms before diagnosis allows us to intervene before somebody loses so many of their beta cells that they require lifelong insulin support.
So one of the things that was really important for teplizumab was sort of that understanding of staging and enabling the regulators to assess benefit risk in stage 2. Carly, I know you and I talked about what you guys are trying to do on the Parkinson side around staging, but then also just in understanding that pre-symptomatic phase of Parkinson's. So I don't know if you want to add on to.
Yeah, so your story is really interesting, and we're not quite there. We don't have a disease-modifying therapy. We're very hopeful that we may be able to actually make better progress in that direction. And the reason is that we now are able to identify a specific biomarker, and that biomarker is a misfolded form of alpha-synuclein, which is a protein we know has been in Parkinson's pathology post-mortem for decades, since the 90s. We've also known that there are symptoms like loss of sense of smell, constipation, rapid eye movement, sleep behavior disorder that precede the motor syndrome of Parkinsonism by years or sometimes also decades. And we now can start putting that together and show that people who have, for example, loss of sense of smell, and we can now measure a biomarker for alpha-synuclein in the spinal fluid, the misfolded alpha-synuclein, those people we can now think of as at that early pre-manifest stage, so before the motor features of Parkinson's are really manifest, and start targeting those people as the very earliest stage of disease where we hope to be able to intervene. So it's a big breakthrough to be able to take these different observations that could only be validated at postmortem and now look at them in living people and start saying, now we can find people in this early stage and think about beginning clinical trials that can target them.
Jiri and Mike on the phone here or coming to us virtually, and Kevin as well. I mean, the three of you guys, we've talked about a lot of other instances of sort of chronic degenerative diseases where these models might be important. So I don't know, Jiri, you've worked in a lot of different areas. I don't know if you want to comment about, beyond T1D and Parkinson's, other chronic degenerative diseases that come to mind that where a framework for how to approach this might be helpful.
And I think what is all common in all of these diseases, and we want to address them in a pre-symptomatic stage. How do we know that the intervention actually it's helpful? Because in all those diseases, I spend a lot of work on Alzheimer's and lysosomal storage diseases and so on. These clinical symptoms which we use for looking at efficacy, they come way later. Takes 20 years somebody to show clinical symptoms. So now we are thinking about addressing the disease in pre-symptomatic stage. How are we going to do that? We cannot do clinical trial which takes 30 years. We're not going to be here after that or it's done. So the question is then using biomarkers and understanding the disease to develop a framework which will allow us to use molecular changes in the disease progression as an endpoint, which we would be preventing of going through these endpoints, right? So, and I think we all kind of understand it in that would be needed, but we really don't have a real framework of thinking that we can actually communicate among ourselves with other scientists, with the regulatory agencies, that what do we need to have to go that route? That I do have an endpoint, which then everybody believes is associated with the disease. If I prevent that endpoint, it's not going to get to the symptomatic stage. How I find the patients and staging is important. So that's what in my mind comes, like, make it practically useful. We need that framework that people can actually work around it, and being able to develop it in each particular indication or in particular diseases will be different, but the framework in my mind will be the same.
Yeah, and I think we'll kind of come back to this framework discussion here toward the end. But Kevin, from a regulatory perspective, what are some of the things that occur to you when you want to think broadly about this idea of pre-symptomatic intervention? What are some of the challenges that regulators are going to face with assessing benefit risk or things like that in this space?
Yeah, so it's a big challenge, and we're seeing a shift now. It's a true paradigm shift to really thinking about a pre-symptomatic population, and of course that brings with it some challenges. So we've heard from patient groups that they're willing to take on the risk and to be involved in those trials to see if it's possible to really fundamentally change the course of the disease before symptoms appear. So some challenges that occur for us from our perspective is really how to show that the drug is effective in that population. So of course, as we heard, that there's an option where you could perform a trial and wait for a long period of time to see if people do form the symptoms and a clinical manifestation of the disease, or you can rely on an understanding of the biomarkers of the disease and the progression of the disease before the symptoms. So I think a key component of that is really to understand that pathophysiological system, and beyond just a single biomarker, but the complete system, what is changing at different time points throughout the disease. So from our perspective, the pathway that we consider typically when we're thinking about a pre-symptomatic population is for accelerated approval, where we approve based on a surrogate endpoint, and then the sponsor will have to do a trial to establish that if you wait long enough, you'll see an effect in the population on clinical symptoms. So really, the challenge we can talk more about is really how would a sponsor be able to develop a story, as we heard in the morning, to show that a treatment is fundamentally changing the progression before you see the symptoms.
Yeah, that makes sense. And Mike, I know you spend a lot of time or have spent a lot of time thinking about this. The idea of intervening pre-symptomatically, and you heard Jiri allude to this a little bit as well, means that we're likely going to be running trials differently. But that also presents some interesting opportunities potentially to move through mechanisms maybe more quickly. And I don't know, just add your thoughts on to what might change in terms of trial conduct or trial design as we move into this new space.
Yes, so the critical elements have already been outlined. First of all, we need to treat the underlying biology and not symptoms as they become visible to a clinician. The work that's required to understand what we could observe that could then become part of a clinical trial setup requires coordinated effort. So for us to deliver and walk the talk on prevention of neurodegenerative diseases, it's likely that we need to bring together basic scientists, clinical trialists, regulators, to form a coordinated effort around an integrated research platform where we try to achieve two things. The first is, how do we appropriately observe patients before they become patients in the eye of a clinician? And second, how can we then use this knowledge to apply the testing of investigational compounds to assess whether we can achieve disease modification? The struggle in the past has been a tension between what we all knew and know is the right thing to do, namely to intervene early versus what is feasible and what is acceptable to the many stakeholders that exist. And I would argue that, in my experience in Alzheimer's, too many times the what is feasible and what has been acceptable at the time won the day, much to the detriment to many clinical research efforts that simply applied interesting pharmacological interventions at a stage that is too late. So I think the time is right for us to work together to create an integrated research platform where we learn through a longitudinal observation of patients as to what the biomarkers or the fingerprints of different biomarkers are, so that we can triangulate the position of a person that might be a future patient along their trajectory, and then use this, say, in a platform trial, where in one integrated effort, we compare and contrast more than just one compound. Now, there are many follow-up questions on, can that be done? It has been done. Oncology is, paving the way, and it's been tried, and it is being done in ALS and the ALS Healy trial. So I think the future is absolutely there for us to make a big splash there. But the critical piece is to agree that we need to treat the underlying biology at an early enough time point, and that we need to find a framework that enables us to do that.
Mike, and you said something, and Josh, you and I have had this discussion over a number of years now in the type 1 diabetes space. Carly, I know it's potentially a challenge in Parkinson's and some of these other diseases. The efficiency with which we can move through some of these, testing these novel mechanisms in this space, ties directly into identifying patients who are pre-symptomatic, the challenges with screening, right? Because people don't have symptoms at this point that announce them as potential candidates for even considering a clinical trial, it means that the pool of people who know that they might want to participate in a trial is naturally limited. So I think maybe what Mike's talking about in terms of maximizing the efficiency of the studies that we can do becomes really important. And I know type 1 diabetes, even though we've had at least an idea of what the staging it has been, and we've been screening for 20 plus years now. Maybe you can kind of comment on that. And I'm just really, 100 years between insulin and teplizumab is an awfully long time. And if that's the pace, we're in trouble. So what can we learn maybe from the T1D perspective? If you guys were going to do this again, what would you do differently? Maybe some of the things you've learned, and when to start.
Yeah, I think to Mike's point, excuse me, this takes sort of the ecosystem working together, right? Drug sponsors, the FDA, patient advocacy organizations, and academic researchers, because there's layers to building that clinical efficiency. Starting out, you have to have a clear understanding of the biology of disease before what we would talk about as clinical onset. So you have to do those natural history studies. A sort of crude analogy I use for this is, if you want to figure out why a car crash happened, you can investigate the crash scene. You can look at tire marks. You can make assumptions based upon the data you have. But if you actually want to understand what went wrong and how to prevent it from happening, having a video of that car crash happening is really helpful. And that's where the natural history and sort of the academic research comes in. And that builds on the ability to actually delineate stages and identifying people as they move through the progression of disease, and understanding that the biology in, let's say, what you call stage 1 is going to be different than the biology in stage 3, which then brings us back to the idea of biomarkers. And I said there's layers here, so I promise I'm getting to the point. But then really identifying biomarkers to enable that rapid clinical assessment is key, particularly for diseases like type 1 diabetes. Running a clinical trial that has diagnosis or clinical diagnosis as an endpoint is not only cost prohibitive, it's also very timely, right? People progress at different stages. There's a lot of heterogeneity, which I'm sure we're going to touch on at some point. But unless you have an ability to have clear markers that somebody is progressing and the rate at which they're progressing, and whether or not they're responding to the treatment, and that gets us back to looking at the underlying, let's say in this case, autoimmunity versus metabolic symptoms, it's really hard to move into that space. So, we were very fortunate to have teplizumab approved with progression to stage 3 or diagnosis as an endpoint. But I think, Joe, you were around during this time. That was a very long trial. And something sponsors have to think about is you cannot just meet your recruitment needs for your trial, because that recruitment for the trial is also going to be your patient identification and your outreach. And if you're struggling to get enough patients into the trial and to move that along, you're going to have a similar problem after registrational approval, because you need to take those biomarkers and put them into screening efforts that actually let people know that they would benefit from the treatment and make the case for why it's important for them to do so.
Yeah, and you brought up this topic of heterogeneity, and I think, all of us have talked about, how that challenges both sort of the biomarker discovery and validation, and then also some considerations about what might be the most appropriate pathway to approach. And Carly, I know you in your research are looking at a lot of trying to understand natural history, root cause, things like that. I wonder maybe if you want to start down this line of what sort of a challenges heterogeneity pose for us, and then maybe we can go to Jiri to kind of comment about how that complicates biomarker discovery and validation, and then finally we'll go to Kevin. Heterogeneity is a problem, potentially a problem for regulators to consider. So maybe we'll start.
Yeah, so maybe I'll start with that, but I also wanted to just mention that the sort of the schema that Mike just outlined is something that has allowed us actually to identify that alpha-synuclein biomarker very efficiently in Parkinson's disease, because 15 years ago, with support from the Michael J. Fox Foundation, we set up a prospective cohort study to identify markers of disease progression. And we didn't have any at that point, and we banked the CSF. And then when at some point people actually became able to measure the misfolded alpha-synuclein in spinal fluid, it was possible to have this systematically collected clinical and biomarker source data, in this case it's spinal fluid, and go back and do those analyses and very rapidly identify this association between misfolded alpha-synuclein and people who had not just clinical Parkinson's disease, but also to look at people that we had enrolled who had, for example, loss of sense of smell as their only abnormality, or people who had rapid eye movement sleep behavior disorder as their only abnormality. See that those people also had synuclein abnormalities. And then, since we'd been collecting data for a long time, that some of them actually then had later progressed to Parkinson's. So it was, an example of having this big systematically collected natural history framework with banked samples that allowed us to then very quickly take advantage of advances in technology. And we're hoping to do a platform trial, sort of as he suggested, to start looking at disease modification very shortly, nested again in that big prospective cohort. But in the context of that, we don't really have great quantitative biomarkers yet until we get to the dopaminergic system. And we would like to be able to not get to the dopaminergic system, but stop disease before we get to the central nervous or to the main symptoms of Parkinson's are dopaminergic, we think. So that's where the heterogeneity comes in, because what we know is that in advance of that motor system of Parkinson's disease and those features, people have loss of sense of smell. Some people have rapid eye movement sleep behavior disorder, but not all of them do. People have anxiety, people have depression, people have, fatigue and pain and lots of sort of non-specific, what we call non-motor features that precede and come before and then come after as well, the motor diagnosis. We also know that changes in cognition are quite variable, and we don't understand the full determinants of that. So some people may have the same alpha-synuclein abnormality and have predominant changes in cognition. And now those people are technically in the clinic called dementia with Lewy bodies, but in fact, it's the same biomarker abnormality. So now we've started conceiving of the disease differently because we can identify a biomarker and say, oh, there's this continuum that maybe starts some people with just loss of sense of smell and ends in people with a motor and or a cognitive deficit. And there's a lot more to learn, but it's, a big step forward. And we've been able to conceptualize sort of a preliminary staging system that maps that out and allows us to say, where do we want to start in terms of thinking about interventions for disease-modifying therapies?
Yeah, so there is an immense amount of complexity. We see some of that in the type 1 diabetes space, and some of it I'm sure we yet to discover even and fully understand. We kind of have a pretty definitive high level understanding that helps with drug discovery. I mean, a little bit different than Parkinson's. Once you have a couple of autoantibodies, it becomes more or less a question of when, not if. So it's fairly definitive fairly early on, but that difference in rate of progression poses a challenge when you're thinking about clinical trial enrollment and who to bring in. So, I think in a lot of these cases, that high resolution understanding, but you have to start someplace. And I think that's a lot of times what maybe holds us back. It's the task is so daunting, and particularly with natural history studies, right? You always get to the point where it's like, I wish we'd started this 20 years ago. And so I think that's something for everybody in this audience. If you're even thinking about starting a natural history study, start with a natural history study. Because no matter what, and technologies have changed, all of that's true, but you do the best that you can and understanding that you'll build on that knowledge and iterate. Otherwise, you're always, well, maybe we should wait another 5 years, or maybe we should wait another 10 years. I think that's a lesson from both Parkinson's and T1D. That's a clear dot that we can connect. Jiri, I suspect this heterogeneity discussion gives you hives.
On what you said, which is fantastic, actually. And that comes to the basic problem or conundrum with biomarkers. And that's a relationship of a biomarker and clinical outcome, right? So that there is a relationship between biomarker and clinical outcome, but it's sometimes imprecise, right? And you see it beautifully in Parkinson's. It's the same thing. It's in Alzheimer's. You've got people, they have it's very well recognized that we have a biological stages of the disease, and then we have a clinical stages of the disease. And of course, with worsening biological stage, you get to a worsening of the clinical stage. But there are a lot of people which are resilient, a lot of people, they are sensitive. And that makes it all mess. It's really, really hard, right? And then, it's difficult if you have the clinical endpoint. That's what you work toward. And then you end up with a huge variability in patients in terms of biology, in terms of the underlying pathology, right? So I think we need to kind of start really thinking more about treating the underlying biology or the pathology, right? Especially in asymptomatic patients. Now we are talking about symptomatic patients, right? And the asymptomatic patients, and establish that link between that biomarker profile and that worsening of the clinical disease at the end, right? But being able to do that, you've got it. Please do so.
Yeah.
Yep.
I've been sitting here thinking about this. This is the holy grail, obvious. But if you think about a disease like, say, psoriasis, and I'm picking it because it's got some well-known targets. Now I don't have to yell. How nice. say, take IL-17, right, approved for psoriasis, but there's somewhere between 20% and 40% of patients who are non-responders, and this is true across multiple diseases. So if you want to pre-treat people, how do you pick what to pre-treat them when we don't even know right now whether they'll be non-responders to IL-17 or IL-23 or like whatever it is? I think this is like just a huge issue in the whole idea of pre-treatment, because we don't even know who will respond today when they have full-blown disease.
So I'm going to aim that. I know, I see, I didn't even have to
Good job.
look. So I'm going to aim that at Josh, and then Mike, we'll come back to you. So Josh, go ahead.
Yeah, it's still something we struggle with in type 1 diabetes. Even teplizumab, which is approved, you have clear responders and non-responders. Our task now is finding biomarkers that at baseline stages, we can identify who's likely to respond. You can't do that from a single trial. Trials aren't powered for mechanistic endpoints. Nobody can afford to do trials that large. So what we're trying to do is to work across the community and to make sure that we've standardized sample analysis protocols that have a clear definition of what a responder is. That's a huge issue, actually. I don't think we talk about that enough. Getting everybody together and deciding what the metric we're going to use for response, what the magnitude of response is required for being called a responder, and then whether or not there's a duration component to that is a big conversation. And it's something we're working through right now in type 1 diabetes.
Let me get to Mike first, and I'll come back
Okay,
to you,
all right.
Carly. Yeah, Mike, go ahead.
Yes, so so many amazing things that's been said, in particular about the alpha-synuclein. Yes, it's true. The underlying biology is complex, and yes, there's high variability. However, the way we currently work is not set up for success, in that we have disintegrated, separate clinical research efforts that on average are rather small. But now imagine to put all of this together and do it in a coordinated fashion, as was just mentioned. I'd like to bring up two examples where that has happened. One example is in oncology, in the breast cancer field. You're aware of the I-SPY 2 trial. There too, it's been unclear as to how to describe the fingerprint, the biomarker fingerprint of patients who would be most amenable to a particular treatment. But it's been the coordinated effort of the integrated research platform that has enabled first learning on how to observe the disease, and second, how to identify the biomarker fingerprint that predicts that a particular treatment works. The second case study, which I think is remarkable, where we can learn a lot from, is the effort from Randy Bateman around Diane. It's taught us that the neurodegenerative clock starts ticking decades before we see something. So that's a given. So we always start too late in traditional efforts. But it's also taught us that we can mature biomarkers into trial tools. So I believe that if this discussion and this CPATH meeting should result in a follow-up effort where we create the design for cooperation between basic biologists, clinical trialists, regulators on how to work together to achieve two things. One, on how to better understand the disease, and two, how to derive from this understanding in the most efficient manner possible a biomarker fingerprint that allows us to then apply investigational compounds against it. Then we're talking. And when you look back at the whole disease-modifying Alzheimer's experience, where we tried out gamma secretase inhibitors, good intentions, different companies in isolation learned the same thing, that there was a family of drugs that all had the same problem. Same thing repeated with BACE inhibitors. Imagine that all of these folks had worked together in an integrated fashion. What extraordinary extra efficiency would have been created? So I would challenge us to create that blueprint on how we can best work together to follow into the footsteps of what's already been said. There's alpha-synuclein now, but can we continue that journey in an even faster way because we cooperate?
Carly, and then Kevin.
Yeah, and so Mike said much more beautifully what I was going to comment in a similar way, but the one piece I would like to maybe add to that is that, Critical Path has been wonderful about facilitating the opportunity for sharing data and making an accumulation of experience and knowledge that can let us push forward. I think one of the lessons from our experience in PPMI is that having banked biologic samples is also amazingly valuable. Your trial failed, but you took a lot of fluid. If we could have that bank fluid combined with the clinical data, and someone else now has a wonderful biomarker, how much faster we could validate that biomarker, or those, I think possibly those multiple biomarkers that will characterize certain individuals who would be best appropriate for a certain intervention.
And somebody like NIH might be really well positioned to help with that problem. Somebody, again, Mike already said it, this is this is a whole of the community kind of effort, with each organization kind of identifying what it can do best to move everything forward. Kevin, I want to come back to you though.
Yeah, I was going to add on, I think in terms of the staging system, it's a way actually to handle, variability. So to start with, if you're going to do a trial, it's important to know that the patients who are in the trial, have the pathology that you're trying to treat. For example, for Alzheimer's disease, a lot of the trials, 10 years ago, 20% of the patients didn't have the target. So if you have a staging system, and you have a technology and biomarkers, that's a way actually to shrink your population and to be more focused. And then second, I think it's to have a staging system, it's also a way to explain the variability. So you had the example of patients who have no pathology or a pretty low pathology, but clinical symptoms are very, strong and severe. That's part of the framework, and those are patients with some kind of co-pathology probably. So that opens up pathway to get, biomarkers for co-pathologies and so on.
Yeah. I think I want to come back to the question from the audience too. I think this is part of the mind shift that you've heard people thinking about, right? So a lot of these drugs have been developed and measured on their ability to sideline symptoms, reduce symptoms, eliminate symptoms. But if we think about disease as a continuum, moving from pre-symptomatic to symptomatic, it's not unreasonable to believe that a drug that can be used relatively early in symptomatic disease might still be perfectly fine just before, a few years ahead of symptoms. Can we derail it and delay it? So I think it kind of comes back to this question of understanding the underlying natural history, what mechanisms are in play at what point in time, and then is there a rationale to bring something like teplizumab, because teplizumab started its development in the new onset space and then came back into stage 2, and then now is being looked at again in symptomatic disease. So it's kind of gone back and forth across that line. But we sort of know from what's been done in T1D that it's a threshold, right? At some point, you've lost enough beta cells that you now need exogenous insulin, but there's no reason a priori to believe that the underlying pathology has shifted over that demarcation line. So that's the shift, right? Don't treat symptoms, treat the underlying pathology.
So that's slightly different. If you're going to pre-treat a patient for, 20 years, but it turns out they're a non-responder to that particular intervention, it's you're asking to treat health, quote unquote, healthy people for a very long period of time when you don't actually know if they're going to respond. So I think it's tricky. I'm not saying we shouldn't do it,
Sure.
but I think it's tricky. When a person has full-blown disease already, you can say, yes, I'll treat them, they'll respond or not respond within a few months. But when you're talking about very long-term, and all diseases have responders and non-responders, I think it gets ethically interesting, as it were.
Well, so Jiri, go ahead.
I just would add to it. Yeah, I totally agree with you. It really depends how well you establish the link and how well you know you were absolutely right, but there are examples and people have done it already, like HPV vaccine. It was approved on a laboratory endpoint, not a clinical endpoint, right? So, but then the understanding of the infection, the hyperplasia and cancer is rock solid, right? So what we need to do is to have a kind of a way how to get to that rock solid understanding that we can use it. And that work toward what Mike is saying, like, so how we actually, work together to do that for individual diseases or individual targets, right? So that's to me, it's a way forward, and that's why I always say the CPATH is a wonderful organization, and I'm sorry, I'm not on commission from them. So to bring people together to have that discussion and actually do something about it.
Well, and if you're treating, I mean, we've talked about this, right? If you're treating to a biomarker that responds relatively rapidly to treatment, then you are going to know who's responding or who's not, right? Because that biomarker is going to change or not, and if it doesn't, then you can probably make the assumption that's a non-responder, discontinue.
And I'm over here. Could I comment a little bit about this discussion on the side of the synuclein space? So I'm Rita Cowell. I'm a professor of neurology at University of Alabama, Birmingham. It's the first time I've come. This is an awesome meeting. So I just wanted to comment on this, the idea of underlying pathobiology as developing from a very early stage, because you're talking about these synuclein biomarkers, right? So like what was mentioned before, if people aren't familiar with synuclein, it's just a protein that accumulates in cells, and people have found that postmortem tissue of patients with Parkinson's, it's accumulated in the brain. So the thought is that could be what's causing disease, and there are genes that cause this to happen. So it's been a very strong area of interest, right? Can we get rid of synuclein to then prevent Parkinson's disease from happening? So that's what Dr. Tanner was saying. You can detect this in people with Parkinson's disease, and so, but what I call that can be more of like a tombstone of neurodegeneration. Even A beta and plaques are things that are at their end stage. They probably start developing kind of middle age, but it's really indicating something is going wrong with a neuron with its normal synuclein, because everyone has synuclein. It's working for you right now as you're sitting here. But it just, at some point, it starts accumulating, forms oligomers, it becomes toxic at some point, and then it somehow is being released so you can detect it. But there is something early on going on that makes it accumulate. And so by the time, even if you were to detect it as a biomarker, you still may have missed that window. So I think just kind of making sure that we're still kind of fueling research on the discovery side is really important. And genetics can inform that. So I'm part of a portfolio for Michael J. Fox Foundation, where we're looking at, it's called the Targets to Therapeutics or Therapies program that they just started, where they're identifying targets, because you need to know what they are to be able to develop drugs, right? You have to know what you want to target in the first place. And they're using genetics as an initial starting point for that. And I think that has a lot of potential. But even in Parkinson's, I mean, we know that it's only about 30 to 40% genetic component to that. So that plays into, the patient's history again. It's like, what have they been exposed to in their childhood, environmental toxicants, anything that even could be autoimmune, right? Related to type 1 diabetes. But I think that's just to kind of reiterate, it's a combination of having to know their history and then probably genetics in there somewhere, something that can, help us, add on to that list. So it's not just loss of sense of smell, right? And not just gastrointestinal problems, but it ends up being more of like a full story for that patient. And I think having this kind of platform for us to discuss it is really important, because it really is like, how can we communicate as groups of people to get full histories on people? right? And be able to then decide if it's worth it to do the test for a biomarker that could, indicate risk. So I just kind of wanted to make comments
Yeah,
on that.
no, I mean, I think it's a great point. And I think one of the things that you said that triggered something in me too, and I want to come back to Josh on this. None of this stops us from continuing to learn, right? Ideally, what we have is a virtuous cycle where we can begin to build. I mean, one of the controversies that's been raging for as long as I've been in the T1D field is type 1 diabetes fundamentally a beta cell problem that leads to autoimmunity, or is it an autoimmune problem that leads to loss of beta cells? And there have been some ferocious debates on that. And at some level, it matters again, depending on what stage you're in, right? So if it starts as a beta cell problem, but then shifts to an autoimmune problem, and that's really the driver of final pathology, then what you want to do for any given person in any given stage in disease may shift. We've got to keep learning, right? So just because we have alpha-synuclein, I don't think that, Carly, that's not the end of the story. That's the start of the story.
Beginning, yeah.
Exactly. But I think that's where we need to go, right? We've been kind of doing this in kind of these one-offs. And I think the people I least envy in all of this, Kevin, are the regulators who are ultimately going to be faced with making benefit risk decisions, ultimately on the basis of biomarkers and the confidence in these biomarkers. And I don't want to forget the safety side of this either, right? We've talked a lot about sort of the efficacy biomarkers. Are there regulatory considerations that if we are going to go into this space, what are some of the things maybe we need to think about on the risk side vis-a-vis biomarkers? Because you also don't want to wait 20 years to see an adverse event pop up.
Yeah, so I think in terms of that side of the equation, I think you also want to be interested in the population, the disease, prevalence. How well do we know if a pre-symptomatic person is going to get the symptoms? Of course, there are some diseases where they won't, they can live and have the pathology and not have the symptoms, where there are others where you're going to have the symptoms. So that is an important factor in terms of how we think about safety. And then, yeah, it's a challenging question. There's no easy answer in terms of how we could to investigate, the safety part of that. And of course, as part of the trials for accelerated approval, you have to expose patients to the drug and follow them for a period of time and still satisfy our criteria for exposures to get the safety information from that population. So the thought is that by the time we get an application, that we will have a lot of exposure of people and who will be followed for a period of time. So at least at the first part, where it's typically where you would see some safety issues arise, it's possible that you're going to catch that. Of course, there's a risk if you wait for a longer period of time that something could pop up. That all has to factor into the equation of patient population, how certain it's going to get, how certain those patients are going to have symptoms, and other factors. Go ahead.
Yeah, and I think with regard to teplizumab, the FDA deserves a lot of credit. There's some creativity that came through in coordination between the FDA, our organization, other patient organizations, and the trialist. In that TN10, which is the trial that was being tested in pre-symptomatic type 1 diabetes patients, wasn't intended to be a pivotal trial. It was really moving into a pre-symptomatic stage to see if we could repeat what we saw in those that were already diagnosed. To the credit of the FDA and listening to the unmet need, and also perhaps the opportunity cost of not meeting that need, knowing that these patients that have 2 autoantibodies are going to go on to develop type 1 diabetes as a clinical diagnosis, took this step of considering the safety data from the previous 4 trials that had been done in those that have already been diagnosed for consideration of the safety in the pre-symptomatic trial. And this really came as a surprise to the field. I mean, we were ecstatic to see it happen, but that trial was never designed that way. It was empowered to be a pivotal trial, which I think is really important. And it speaks to the creativity that can come from all of us kind of working together to meet what we define as a clear unmet need for patients.
That's a great example, Josh. And some of those trials were, quite long in the tooth. But in aggregate, having that safety information, at least in terms of, and in some cases, a little bit longer follow-up for some of those folks or ability to reach back out. I think a safety package that the FDA was comfortable with taking some risk then on the efficacy side. I think that's an important point, and it kind of comes back to the earlier comment. We probably do have some drugs that can work in the pre-symptomatic space, which we have a pretty large safety profile of. And maybe that, I don't want to put words in your mouth because it's always kind of going to come down to the specific drug and case, but all of that goes into benefit risk considerations and may enable, the regulators to take some more risk in an approval when they have comfort on the safety side, if the unmet need and everything else is there. Mike, I see you've got your hand raised too, so yeah.
Yes. Hey, I wanted to come back to the comment from the audience just now on alpha-synuclein. I totally agree, but what an achievement. If you think back 25 years ago, we had the odd paper that we had read, and if we were arguing to run a clinical trial about this, no way we would be allowed to do this in a pharma R&D environment in the 90s, in the 20s. And so it's an advantage. But here's my main point. I think we all work on the same goal, and that is to serve patients, to create better solutions for the future patient horizon. But there's one thing that needs to change dramatically, and that's the approach that we take. I'd argue that the way we currently work, we think about different stakeholders. In isolation, we talk about the regulators, the clinical trialists in a pharma R&D setting, the patients, etc. This is all one community. And when I look at big projects that really have moved the needle, Manhattan-like projects, with a good objective, there's a clear existential goal. We have that goal. There's a top mandate and a sense of urgency. There's funding. Look at what is happening in Parkinson's, just extraordinary. There's so many elements here together. But then there's centralized, agile leadership. Central, agile leadership. And there is that clarity around what success would look like. I would argue success looks like if we don't have to wait for 40 years or 30 years, as it took before we agreed that lowering blood pressure was preventing death from stroke or myocardial infarction, or 20 years before we agreed that minimal residual disease is a good surrogate for in the field of multiple myeloma. But if it takes 5 years for us to work together to have solutions that will ultimately enable prevention of neurodegenerative disease. But for that to happen, we need to come together in a proactive way to have clarity as to what the call to action is, who is in charge of leading this, and how we can maximize the efficiency of knowledge creation. We all want the same thing. We are one community. What should we do next? My suggestion: to create a blueprint for such a big project that lays out exactly what it would take for us to come together across the different angles from which we look at the problem.
Yeah, and I think that's the perfect segue before we go to some audience questions here, Mike. And we've all talked about this. I think in all of our discussions, what we've realized is there's no consistent framework that guides this. And obviously, there are going to be disease-specific considerations. But even at the high level, okay, what do you need? What do you need to think about? What do you need to be collecting? How is that data going to be aggregated? How is it going to be used to enable regulatory decision-making? These are all questions that we're kind of trying to solve in individual buckets. We're not connecting the dots. And there's a lot of experience and information out there. And so I think what we all kind of came together and decided is that one of the outputs from this meeting, and Klaus mentioned earlier, this meeting's not about the meeting, it's about what happens and then what we can say the next time we get together, is we need this framework. We're going to need the input from the people who have experience, what they've faced as challenges. Can we replicate success and build on that? What does the framework need from a regulatory perspective? Ultimately guidance. some sense of, from a drug developer's perspective, that we kind of know what's going to be needed, what types of evidence we're going to need to generate. That's what will enable the investment, and that will only build upon itself. Another thing that we've talked about is we know, okay, these neurodegenerative diseases aren't as distinct as maybe we thought they were. It's certainly true with autoimmune diseases. What can we learn across the board? Because these things tend to live in silos. That data's out there. Those samples are out there. Carly talked about the biorepositories. We need to do a lot better job at connecting these dots and then moving into this pre-symptomatic space. But we need to do it, as I said at the outset, in a consistent and determined manner, following the roadmap and adding details to that as we go along. But this is the shift from doing these one-offs, disease by disease, and taking it as a framework that can enable replication of this approach across the board for these chronic degenerative diseases. So everybody should. I'll put at least myself on the hook. I don't want to put my colleagues on the hook, but Jiri, you want to?
This is exactly right. So what you are saying, 100%, and there is also a precedent for it, because we've developed. I mean, a couple of years ago, we developed a framework for biomarker qualification as a group, under PSTC and with collaboration, bringing the stakeholders together, the academics, the labs, the FDA, everybody, and develop, and then ended up actually with the FDA guidance. And that framework, it's high level. It's kind of like what you need to do, kind of to get there. And I, as a sponsor, I used it many times. It's for not biomarker qualification, but for discussion about, I've got this biomarker, when I use it on my trial, and I use this framework with the regulators, and we all knew what we are talking about. And it was really, really, I mean, it's helpful on many levels. So I'm with you.
And the other element I don't want to lose, because we've had this conversation many times over the past day and a half, is who's not in the room. Payers need to be part of that conversation, right? Part of that framework is the evidence generation that enables them to understand what reimbursement needs to look like. And I'm sure there are others. So I think we need to be comprehensive and think all the way to the end, which is effective drugs getting to patients. And everybody that's on that pathway needs to be part of this conversation ultimately. So I think with that, I want to open it up to the audience for engagement here. So I think we'll start. Yeah.
So Derek Hill from UCL and Panoramic Digital Health. So I'm trying to triangulate in my head between three things: this panel discussion about prevention, biomarker-based evidence, the previous panel about patient-focused drug development, and also the reality of anti-amyloid immunotherapy coming on the market, approved by really quite brave decisions by the FDA, initially ahead of other regulators, but yet not being initially anything like as successful in terms of patient adoption or clinical adoption as, for example, the Alzheimer's Association hoped. And I think the issue here is that it's quite possible that we won't get biomarker signal change dramatically, resulting in complete halting of disease progression. We will get something which is much more nuanced. There will be side effects of the drug, maybe like area we've got in Alzheimer's immunotherapy. And that, I guess that means that the benefit risk to the patients is much more nuanced than it is with statins for treating risk factors for cardiovascular disease. And I guess I don't feel this conversation has really wrestled with that reality. If I can be bold, it feels a bit like conversation I heard like 15 years ago, or 10 years ago, when people were thinking about, getting Alzheimer's drugs to market, and it didn't turn out like people hoped. So again, I'm just quite interested in your perspective on that perhaps slight disconnection.
Yeah, I mean, I'd love to hear from the panelists. My perspective is that a lot of that comes down to the certainty or how well you really understand the underlying biology and how well you can link the biomarker to that pathology. So undoubtedly, we're going to miss some things along the way. It's not a reason not to go in, but I do think your comment about, again, it comes back to that benefit risk equation for the patients. But Jiri, yeah, go ahead.
I would add to it that specifically for the anti-amyloid, right? We're on the beginning of actually learning how to use them, and I personally think that there is a patient population which would disproportionately benefit from them, and we don't have it yet. It needs time, and there are studies coming in now in pre-symptomatic, earlier and so on, and we'll see how that goes. But it just needs the natural evolution. I think the huge step, I agree with you, to have those drugs actually out there and to be able to learn, how to use them and stuff, and then develop a new generations out of it, right?
Joe, can I jump in? Because Derek, what you said resonates on one hand, but I want to remind us of a total unforced error. There's a ton of stuff we do know very well. So we know the postmortem studies. They've been written up, I don't know how many decades ago, but we ignore what's coming out of them. It's damn clear that we need to intervene early. That's not news. But what do we do when we translate this from a feasibility perspective to achieve disease modification? We sort of fiddle around and come up with an excuse. In other words, I would argue there's a lot that's already known, but we can't bite the bullet and argue, yes, it's difficult, we'll still do it. I feel that is a much greater risk than the thing that we don't know about that also might come about, but we'll learn about it. So the unforced errors that come from not doing what we know, I feel, is the biggest risk in this field.
Right. So good morning. My name is Ralf Herold from the European Medicines Agency. And to your point, I think there will always be more questions, and that's the challenge that we had. I would like to come back to the aspect that there are cross-cutting methodological issues that cut across the therapeutic areas. I know it's about Parkinson and diabetes. I want to add that recently we started in the European Union funding a large consortium. It's called Intercept, appropriately, but it is in inflammatory bowel diseases, and the long title reads Verification and Validation of a Biomarker Panel that Identifies Individuals at High Risk to Develop Crohn's Disease, Allowing Interception of Full-Blown Disease Development. And there is now a period of 5 years which we can use to coordinate between CPATH activities and between these and maybe some other activities through a little bit getting the perspective, Mike, that you called a blueprint. We can find the right words to address these cross-cutting methodological issues. Josh, you posed the challenge, how will the trials look like? So if we don't know yet, let's work on that. It cuts across, and there will be models of discussion of choices being made, etc. But the point is that we need to facilitate these cross-cutting discussions between the projects. Otherwise, we have or may not have an IBD solution. We may have a solution here and there, but the underlying principles, the challenges, the long duration, the uncertainty, decision-making, etc. There's the ethical questions that were raised. They are not unique. And I just want to pitch this here, and maybe it's more a comment than a question that you can take as it is, and my willingness to facilitate that. But please, it has to happen globally. This is the same thing, the same science, the same ethical questions. So let's look at the therapeutic area, sort of independent methodological cross-cutting questions.
Could not agree more. And Josh, I don't know if you want to make a comment on Identify, which is another large effort going on in Europe related to screening for pre-symptomatic type 1 diabetes. I know you guys are involved in that at some level. So
not much to add to that other than, these screening efforts across various geographies are critical to all the things we talked about understanding before, right? To getting a good idea of exactly what's going on in different populations, different backgrounds. I think, one of, not to sort of turn this to a negative, but one of the barriers that we have to work on with programs like Identify and Intercept and things like that is, making it easier to share samples and data across geographic borders, so we can actually reach the power we need to pool this together and to actually identify biomarkers and improve staging and all of these paradigms is a critical part of moving forward. I feel like sometimes it's a little bit like driving a Ferrari through Jello, right? We have all the momentum, all the speed, but we're just not moving as quickly as we can to get things to patients.
Yeah,
We
and
have done this for minimal residual disease, for example. There were similar
Yep.
efforts in the US. And so I think there's a very good starting point. So the next 20 years, in a way, can
It's
deliver
another
more.
dot to connect, right? I mean,
Yep.
this is exactly what we're trying to understand. But I love this idea of working with Intercept and pulling that into this equation. But yes, right, Carly, go ahead.
So an additional comment, sort of harking back to the prior conversations is, you know, what happens when a drug comes to market and it's not the wonder that you thought it was going to be? I mean, I think, first of all the comments to that were appropriate, but I think it also highlights we need to have a mechanism for understanding what happens when a drug comes to the community. There always is a difference between that perfect clinical trial population and then the rest of the world, the ordinary people, when you bring a drug out, and it varies in terms of how much difference there is. We need to understand that. We need to try to understand more about how can we systematically be aware of those changes and fold that into our blueprint as well, so that we can learn to do better and to understand the individuals who are the ones who are going to benefit and those who shouldn't be. So, you know, getting closer to, quote, precision medicine and figuring out who are the individuals who will benefit from one specific treatment. So I think that's hard, but trying to think about having that be part of our blueprint, if you will, I think could be really helpful.
I really apologize. The boss is cutting me off.
Joe,
Well,
before
listen,
you
thanks, everybody. I really appreciate the audience engagement on this topic. And it's clear there's a lot of work to be done, but the sooner we start, the sooner we can finish. So thanks.